Hello, good afternoon, everyone. My name is Jaiwante Samsoondar. I'm an agricultural officer at the research division. I'm sorry, I just got a little bit of feedback there. Yes, and today I would like to thank you all for spending time, your lunch hour with me, where we're going to go through the research activities being done at the Cocoa Research Unit of the Ministry of Agriculture in the Research Division. So before I start into our work plan and what we've been doing, first of all, I'd like to say that our work plan is developed in collaboration with the major stakeholders of the cocoa industry. So they have a number of committees, and in these committees is comprised of the Cocoa Research Center, the Cocoa Development Company of Trinidad and Tobago, as well as all the regions, Region Admin North, Region Admin South, and the Agri-Services Division of the Ministry of Agriculture. So it's a body of people comprised of varying experiences that come together, and we plan what research activities we're going to do. So the format of my presentation today is a PowerPoint presentation where we are going to go through, my PowerPoint is stuck now, where we're going to go through Mr. Ramdas, I'm having a little bit of technical difficulty. Right. So it is just sticking. It's a bit slow. Forgive me. So the key areas of focus that we do within the research division is genetic improvement through breeding. We have a couple of agronomy trials. Over the years, it changes. And currently, we're looking at cadmium mitigation because cadmium is a very topical issue within the cocoa sector these days, as well as at the La Reunion Estate, we focused on cocoa germplasm conservation. Now, for some of you who are familiar with the ministry, you would be aware that La Reunion Estate is the home of the TSH, which is Trinidad Select Hybrids, the breeding program. Our breeding program would have started in the 1950s, and it continues up to this day. Currently, we have breeding taking place, and it is considered to be one of the most successful in the world, and is uninterrupted. The initial goals were aimed at ceratosis wilt, and which is broom disease resistance. So in the 1950s, when it started, the whole breeding cocoa program started with the history of cocoa in Trinidad, where we had Criollo type cocoa, and then they started to die, and then Forastero was introduced. And then what they found is that there was natural hybridization between Criollo and Forastero, and that's how we came up with Trinitario. Now, what happened is that from that natural hybridization, the Imperial College of Science, they would have selected 100 crosses, which we call the ICS crosses. So the ICS crosses is that natural hybridization. And then from there, what we did was we introduced new genetic material, and we started to introduce genes for resistance to ceratosis wilt, and which is broom disease, and that's how we came up with the Trinidad Select Hybrids. And currently, that is predominantly planted by the farmers in this country, TSHs. Now, the persons who are familiar with cocoa will be aware of the naming of the TSHs. So when you go to purchase plants, you would hear TSH 919 or TSH 1188. So how did this nomenclature come about? Now, the large number of progenies, because remember, our breeding program is advanced by hand pollination. So what we do is we introduce genes from a male parent to a female parent by hand pollination. Now, that hand pollination, you take the flower, and the male flower, and you pollinate the female flower. You cover the flowers, and then you wait for the pod to mature. Now, when the pod matures, what happens is every seed within that pod is unique. So what we do is we plant out the seeds, and we assess the seeds the plants, seedlings in the field, and that is how we select to move on in our breeding program. So the early scientists, they started with the prefix TSH, which stands for Trinidad Select Hybrid, and then is followed by a number which runs consecutively from 500 upwards. Now, currently, the commercial TSHs, the earliest one we have that's available to farmers is TSH 730. So even though we start at 500, not all of the plants that are planted out and assessed make it to commercial stage. How do we select what goes into commercially for production? So the initial goals of our breeding program were aimed at a large bean size. So for those of you who are familiar with the TSHs, when our beans are dried, one bean weighs approximately 1 gram. So that's actually pretty good. And chocolatiers love that because what it means is a large bean. And because you have a large bean, it means that there's less shell because you have the bean to shell ratio is different. Smaller beans, you'll have more shell. And in the chocolate making process, the shell is discarded and all you use is the nibs. So a large bean is actually highly sought after by chocolatiers. The TSHs are early bearing. They have a low pod index. Now, a low pod index is associated with a high yield potential. So what a pod index is the amount of pods required to produce 1 kg of dried beans. So the fermentation process, we have a conversion ratio, and 1 kg of dried beans is after you ferment and dry. Most of the TSHs have a pod index between 15 to 20 pods, which is very good. The maintenance of the traditional fine or flavor status of Trinidad and Tobago cocoa. So all of the cocoa in Trinidad and Tobago is classified as fine or flavor. Now, those of you who are not familiar with the chocolate industry would probably wonder what this means. And it's very simple. In the cocoa world, we have 2 types of cocoa. One is bulk cocoa and the other is fine or flavor cocoa. What's the difference? Now, in bulk cocoa, which constitute the majority of production, when you taste that chocolate, all you get is a basal chocolate flavor. Whereas when you taste fine or flavor cocoa, well, at first you get the chocolate flavor and then there are additional notes. So it's like you taste this chocolate and after you finish, there's still an orchestra playing notes within your mouth. So you would get fruity, you get floral, you get spicy, you get nutty. And these sort of things are what makes our cocoa being sought after in the entire world because of that fine or flavor status. So our cocoa sometimes is used by the artisanal people to make specific country or estate-specific chocolates, or is blended with other cocoa to introduce that flavor. Our current breeding projects, we have 3 projects currently that are going on. The first is the registration of the TSH 1300 series with the Intellectual Property Office. So what the ministry has done is that we have filed for 11 types within the TSH 1300 series, which was the last set of commercial cocoa varieties that was selected for intellectual property rights. What this means is that if we sell the plants or we sell the chocolate and the genetic material, we will get royalty as a country for these material. So it will facilitate trade of the material. Currently, we do not trade our TSHs. We do not sell planting material anywhere outside of Trinidad and Tobago, even though there have been reports of it being elsewhere, but is not sold as a country to other countries. The other two projects we have is that they're both geared towards improving disease resistance. Now, in Trinidad, we have Blackpod disease, which is a very common disease, and most farmers suffer from it in their fields, and it can cause losses up to 60%. So what we've done is, I'll speak up to it in a little bit, we've introduced new genes from the Cocoa Research Center's gene bank, which will assist in providing a higher level of resistance to Blackpod disease. And our newest project is improving the resistance of the TSHs to Frosty Pod Disease. Now, we do not have Frosty Pod Disease in our country. It's been reported about 5 years ago in Jamaica. It's present in Venezuela. So we believe, even though we're trying very hard, our quarantine unit tries as best as they can to keep the disease out, but we are aware that it is likely to enter our country. And as a ministry, we have started preparing mitigation strategies to deal with this frosty pod if and when it eventually comes to our shores. Now, as I said, we have 11 TSH 1300s, which we would have filed for intellectual property. This is the list here: 1362, 1330, 1352, 1315, 1356, 1350, 1347, 1380, 1344, 1313, and 1364. Now, as you realize, the 1300s would have run from 1301, well, from 1300 to 1399. But from that 100 that was selected, these are the only ones that would have shown the best commercial properties for selection, and that's what we're moving forward with. Now, Blackpod disease, this is a photo of it here in a really bad situation where a lot of the pods were not harvested and the pods of the trees really infected. It's caused by Phytophthora palmivora. It's a fungal disease, and it is very prevalent at the onset of the rainy season. It occurs throughout the entire length and breadth of Trinidad and Tobago. The loss potential is up to 60% of the yield, and the mode of spread, it spreads by rain, ants, insects, rodents, and from contaminated prunings. Now, how do we recognize Blackpod disease? Is that it's actually a brown, on the pod, when you look at it, you'll see that it's brown. It starts on either end, and it smells like fishy. There's a fishy smell. When you crack the beans, you will see the beans turning black. Now, this is an issue which we've been dealing with farmers and fermenters alike, because depending on the stage that you harvest the pod, if the black pod is now starting, many farmers still use the beans because the inside of the beans, they still look okay. But because it's a fungal and we don't know the level of infection that would have been within the beans, we advise farmers not to place black beans, diseased beans in their fermentation mass. So I'm just waiting for my slide to change. Sorry. Right, there it is. So in 2011, we started a collaborative project between the Ministry of Agriculture, Land and Fisheries and the Cocoa Research Center, which is the home of, which has the responsibility for the International Cocoa Gene Bank of Trinidad and Tobago. They're at the university, and the gene bank is right here at La Reunion. And what we did was we introduced new genes from the Cocoa Research Center has a germplasm enhancement program, which is the GEP, and we use those the pollen from those trees or those varieties which were shown to have the highest level of blackpod resistance, and we bred it into the TSHs. So basically, what we have are crosses between some of the better TSHs and those varieties with highest resistance to blackpod disease. Now, 30 crosses were tested. So as I said, when we do the pollination, so we wait for the pods to mature, we harvest the pods that were pollinated and we grow them out in the greenhouse. Now, when those crosses were grown out, what was done was a leaf disc inoculation. So the leaves were harvested and they were inoculated with blackpod disease. And based on their reaction to the inoculum, those that were highly resistant, they were planted out. So we didn't plant out all 30. We're not assessing all 30 crosses. So out of those 30 crosses, 10 of those crosses were identified as having high resistance to blackpod disease, and they've been established in a field trial. Now, the field trial is currently being evaluated for selection of the best performing crosses before it can move forward to the next stage of the breeding program. What has to be done is once we ascertain the yield potential, the disease resistance, and as well as the final flavor attributes, those varieties will be selected and they will be planted out in an agronomy trial where it will be assessed before it is released as a commercial variety. Now that will lead to our TSH 1400 series. That's where we are right now with respect to blackpod disease. The next project we have in breeding is for Frosty Pod disease. Frosty Pod Rot is caused by the Basidiomyces Moniloptera rori. It is found in all northwestern countries in South America and now Jamaica. However, it is not, as I said, present in Trinidad and Tobago, and it has and it can devastate our industry. So at the ministry, we have a number of approaches that we are undertaking to mitigate against Frosty Pod. What we have is we've bred some disease-resistant genes into our TSHs, similar to what we would have done with the Blackpod trial. That is only one of the approach we're taking. Every year, our crop protection unit at the research division, they undertake a nationwide survey where they look for the presence of Frosty Pod disease, and in the event that it is found, they have developed a plan, a strategy, which will come into action to deal with the sighting or the presence of Frosty Pod. We also have at the research division what we've started doing, and it was a recommendation from one of the committees, is that we've started introducing ICS 95, which has been found to show some level of Frosty Pod tolerance across the country. So at research, we would have set up a budwood garden, and that garden is accessed by the Agri Services Division, and they would take cuttings and sell, they would propagate the plants and sell those to farmers throughout the country. And this is also one of the strategies which we have in place as Frosty Pod mitigation. Now, this is, I kind of ran through that. So in addition to disease breeding for disease resistance, we do have some agronomy trials in the past. Those of you familiar with cocoa would be aware of the arboricide trials, spacing trials, and that sort of thing. Currently, we have 2 trials that are going on. One is a fertilizer trial, and the other is the dwarfing of the TSHs. Now, this fertilizer trial was done last year, and we are in the process of analyzing the data. Now, what we realized is that in about maybe 3 or so years, there was a survey done as part of the impact project by the CRC. And what they found that the average levels of 150 kg per hectare was being obtained by farmers, whereas the potential yields of the TSHs is 1,500 kgs per hectare. What contributes to this? One of the things that contributes to it is poor fertility in the soil. So the objective of this fertilizer trial was to investigate the effect of fertilizer on cocoa yields. What we did was, as we all aware, if you have shaded trees or overshaded fields, your fertilizers will have no impact. So we started the trial by the pruning of the fields, which coincided with the onset of the rainy season. Now, after the field was pruned, we applied fertilizer and limestone. We just used a split plot design. In our experiment, we had 6 blocks, so we fertilized 3 and we left 3 unfertilized to compare the yields. What we did after, what we noticed is that after the fertilizer was applied, we had a crazy amount of flushes on the tree because it took up the nutrients and it responded by vegetative growth. So what we did is that we went through and we removed the vegetative growth, we removed the flushes from the tree, and then when it started to bear, we took harvest data. So we collected yields where we cracked the pods and measured the weight of the beans, wet bean weight, which would be converted to dry bean weight for data analysis. Now, everybody in cocoa will tell you that TSHs are very, very vigorous plants. It grows like crazy. So the dwarfing trial was actually recommended again out of one of the committees, where leading back to that yield potential being so low, it was recommended. One of the reasons why, in addition to poor fertility, that yield potential is low is because in order to get yield out of cocoa, you need to prune the trees. And many farmers do not have a rigorous pruning regime. So they feel as though you prune once and that's it, but pruning is something that's required to be done every single year because these trees are very vigorous, and once that canopy closes, you don't have proper light, no airflow within your fields, and it doesn't result in good bearing of the trees, and those pods that do bear, they're very much diseased. So if it is that we are able to produce a tree that's shorter and less vigorous, it would be to the benefit, it would require less maintenance by the farmers. So what was recommended was that we use a slower growing variety, and we try to graft the TSH onto that rootstock to try and make field management easy by having a smaller tree. So this process was very, very long as well. When you're dealing with tree crops, everything takes very, very long. So what we did was the IMC, which is Iquitos mixed caballeros, was identified by the Cocoa Research Center as being a relatively small tree, slow growing. So what we did was we hand pollinated the Iquitos mixed caballeros to produce pure seed. Now, 5 to 6 months later, we harvested those seeds, those pods, and we sowed the seeds. Now, we are using TSH 919 rootstock as a control. Now, when the rootstock was of age, we grafted the 10 commercial TSHs to both rootstocks, and we in the process this year of planting out the field to evaluate this dwarfing potential. Right. So what we move on to next is cadmium. Sorry. Now, cadmium is a very topical issue these days. It's been so for maybe the past 10 years, and there is a great concern throughout the world about cadmium in cocoa and the transmission to its derivatives, since cadmium has been identified as being deleterious to human health. It is a toxic metal and it accumulates in your body, causing damage to kidneys, skeletal and respiratory system, and it is known to be carcinogenic. Levels are generally not set on those foods which contain the highest levels of contaminants. Excuse me. But with those which contribute the largest portion to our diet. For example, the brassicas are actually high accumulators of cadmium, but these restrictions have been placed on cocoa. Forgive me. Now, this maximum levels, there are a number of different food safety bodies that have enforced these levels. The first is the introduction of Regulation 488 in 2014 by the European Commission, which sets the maximum levels of cadmium in a range of foodstuff ranging between 0.1 to 0.8 milligram per kg in cocoa and chocolate products. This regulation was effected on 1st January 2019. Now, if we look at the dates, the regulation was proposed in 2014, but it was only effected in 2019. And the reason for this is that there was a lot of lobbying by the Latin American and Caribbean countries against this introduction of this cadmium, because believe it or not, it's only the fine or flavor countries that have this problem with cadmium. It's not where the bulk chocolate is produced. In 2018, the Codex, which is another big food safety body, Codex Alimentarius, proposed maximum limits of cadmium in cocoa products. And in 2018, also, the state of California introduced what we call Proposition 65, stating that cocoa products exceeding these limits should carry a warning, like what you see on cigarettes, a warning label that it may contain materials which can be cancerous or deleterious to human health. Now, cadmium mitigation, this is something that the ministry is working very, very closely with the Cocoa Research Center. So given that we build on our capacities, so we have the fields and they have the laboratories, so we work hand in hand to determine some of these things. What we're looking at is determining cadmium bioaccumulating ability of the TSHs. So we're looking at all the commercial varieties and we want to see if it is that they accumulate cadmium differently. We determine the relationship between bean and cadmium levels. We're looking at cadmium content between fermented and unfermented beans. We're looking at seeing whether the cadmium levels varies along the tree. So a pod harvested along the trunk, will it have a different cadmium content to that of a pod harvested on a higher branch? And that will determine your management strategy, how you would prune. We're looking also at seeing if there's a seasonal effect on cadmium uptake. Now, this has been going on since 2020 into 2021. We're currently awaiting laboratory analysis for the 2021 samples submitted, but the initial first year would have shown that unfermented beans were approximately double that of fermented beans. So unfermented cadmium levels range between 0.8 to 3.85, whereas the fermented beans range between 0.7 to 1.89. At the La Reunion Estate, the research division is very, very focused on germplasm conservation, being the home of the TSHs, as well as being part of international projects. So we have a number of conservation trials. We have the International Clone Trial, the Local Clone Trial, the Regional Varietal Trial, Observation Plot, and the 1300 series. Now, we would have established this ICS 95 budwood garden, and you'd see here they're taking cuttings for the propagation station to do work. We have the Germplasm Conservation Local Clone Trial. This trial was established in the year 2000, and it consists of 20 TSH clones. We have the International Clone Trials, which has 21 international clones. And of course, we have the Regional Varietal Trial with 15 cocoa crosses from Latin America countries. And in addition to the work that we have ongoing, we are also heavily involved in PSIP, which is a Public Sector Improvement Program. Our project is called PSIP J426, Rehabilitation of the Cocoa Industry. Now, as we all know, cocoa has been identified as a strategic crop, and we have been classified as 100% fiber flavor cocoa. Now, again, what that means is that we get premium prices on the world market for our cocoa, and this PSIP is a bit different from our regular work program in that it focuses on strategic interventions to increase yields, resistance to disease, and meeting international regulatory requirements as it relates to heavy metals. Now, the technology that we look forward to in our PSIP is aimed to increase productivity from 1 kg dry beans per tree to 3 to 4 kgs dry beans per tree, thereby increasing farmers' production and, of course, their income. We have 4 activities within the current fiscal year that we are focusing on. One is breeding. The second one is cadmium mitigation. The third one is we're looking at establishment of a solar-powered drip irrigation system. And of course, the last one is establishing a GIS system for management of cocoa germplasm. In addition to, we are the research division, so we don't really do outreach like the extension division, but what we do is that we work with the extension division as well as the cocoa development company to provide technical assistance to farmers as they require. So we are often invited to do training, and we would work with everybody who needs assistance. So that would bring me to the end of my presentation, and we will open up in a little bit for questions. But before we go, I'd like to thank Dr. Rabindra Ramjatan for all of his assistance today. So thank you. I'm going to share my screen. Right, so I now invite you to send any questions that you may have for us to interact, and we could have a live one-on-one session. Any questions? Mr. Ramdas? Okay. If you all, I realize you all don't have any burning questions right now, but if you all feel as though you, after the presentation, there's something more that you want information on, please feel free to contact our technical officer. His name is Mr. Deodat Sigobid, and his email address is ces@gmail.com. Okay, so I'm getting a question from my boss here. He's saying that someone asked a question, where can we get plants for sale? Okay, so plants are available at the Agriculture Services Division, and it is located at La Reunion, and it is at a cost of $2.50. So you can call first, ensure that they have plants before you come to purchase. I'm seeing here for land, what's the procedure to apply for land? Now we have a land management unit at the ministry. They're located in El Socorro. So the procedure would be to go to them with respect to land. Sure. No, say it because the nursery doesn't sell plants. Okay.
What strain should I plant first, or is it based on what they have?
Okay, so I'm seeing another question here. With respect to the variety that you plant, when you go to purchase plants, what is recommended is that we have 10 commercial varieties as produced by the Agri-Services Division. Now, cocoa plants are available at La Reunion Plant Propagation Station, and to some extent the MAPA section in Plumitan. So what is recommended is a mix. We have what we call pollination combinations. There are 10 combinations. There are 10 plants pairs that is recommended. So when you go to purchase plants, you make sure that you get a pair that is on the recommended list. And how you would know is that the Agri-Services Division will have the list and they can tell you what to purchase. Now, Tobago farmers access plants in the same way that Trinidad farmers access plants. We've had a few Tobago farmers, what they do is they send across their transport. So to make sure that their plants, the last shipment that went to Tobago, they would have written to the Director of Agri-Services, Mr. Alan Balfour, and he would have made the necessary arrangements through the La Reunion Plant Propagation Station for them to come and purchase. The plants are loaded onto their vehicle, and the vehicle then goes onto the boat and the plants are taken to Tobago. That's how it works right now. You can also call the plant prop station 642-4188 to ensure that there are plants available and when and how you can collect. Okay, so seedlings. The question is the information on growing seedlings. Seedlings are not recommended by the Ministry of Agriculture to be grown. The reason being is that we've found seedlings to be highly variable. So some seedlings will do very well, whereas others will have very, very low yields. So we only recommend grafted plants with the 10 commercial varieties, or clonal plants, again, with the 10 commercial varieties. Any other questions? Okay, so thank you very much, everyone who would have joined us on their lunch period for this session. I hope it would have fed your thirst and your curiosity for what is happening at the research division with respect to cocoa. And it's rather sad that it's a virtual session. Otherwise, we could have had some nice chocolates to wrap up our session. But I would like to encourage everyone joining in that we should support our local chocolatiers. We have some excellent chocolates that are made right here. And please patronize our local chocolatiers. Thank you, and everyone have a good day. Bye.